Hi everybody, I'm Irene Borra Serrano, researcher at ILVO, Belgium. Would like to start this presentation with this picture that you can see in the slide correspond to a breeding trial, and different genotypes of grass are established there, and our breeders score or evaluate some important or relevant characteristics for a further selection. As you can imagine, this task is time consuming because in those type of trials, there are really hundreds of plots to evaluate. And also, it's not just one trait, but several of them. So you need to revisit the field at different times of the year and also over the years. It can also looks like it's kind of easy to find differences. There in the image, you can see already that there are some genotypes higher than taller than others. But when you have to do it for hundreds of them, or also look at traits such as coverage, or it can be a really difficult task. So I would like to talk to you today about phenotyping, but using a drone and instead. First of all, I would like to start defining what plant phenotyping is to evaluate the performance of a crop based on desirable characteristics to face challenges such as climate change or to increase the production. There you can see in the slide an example of two different plots of grass, and you can see that one covers the soil more than the other one. So you don't see the soil as or the different lines of the crop. So this evaluation of the crop can be done visually then, because you give a score. You can also do measurements. You can also measure the height of the crop, and you can also take samples, so destructive sampling. But these methodologies have some constraints that I would like to talk to you about. First of all, difference in criteria. If two breeders maybe can probably or can give different a score to the same plot. So there is some difference or there is some difference there. So human biased. Then also in those type of trials, you have hundreds of plots, as you can see there in the image. Uh, each rectangle corresponds to a genotype that needs to be evaluated. So it takes a lot of time, so it's time consuming. And also the different selection that needs to be done along the years takes a lot of time. So what do we want to achieve is to have a methodology that is fast, that we can cover the entire field, so every single plot in a shorter matter of time. So with a drone, it's possible to do it in minutes. So you can, in less than 10 minutes, you can cover the entire field of around half a hectare, and it, it takes way less time than, for example, when a breeder needs to go to the field and score each individual plot in the field. That takes hours. So we want also to have a method which provides us with detailed information. If we compare the information that we get when we measure with a resin plate meter once per plot, so we get we just get one value for the entire plot. Okay, if we measure 2 or 3 times, we will get 2 or 3 values, but it's more time that you need also to take those measurements, so the time increase compared to the drone when we can have every single pixel value inside a plot. So it's way more detailed, and also we can capture the variability within the plot. And why a drone and not a satellite imagery? Because these remote sensing products are also free and available. It's a matter of resolution, of detail. Our plots are small regions. It's not a big form film. So we need detail, and we need to be able to differentiate the different plots established in the field. So that's why we tend to use a drone that provides us that high detailed and high resolution imagery. And also this method or this tool allows us to go as frequent or as often as needed to monitor the crop, and we can see the evolution of the crop, for example, here in terms of height, but in the different flights that are carried out through the growing season. And how this is done, first of all, we have to design the flight plan. Depends on the trait that we want to measure. We are going to decide about the sensor to use, the altitude, how frequent do we need to go to the field, and the timing. When the weather conditions are okay to go to the field and the moment arrive, you go to the field, you fly over the trial, you need to really be sure that you not only cover the trial, but also some borders to have enough overlap of images in the entire field. And you need to be careful when in the settings of the sensor and to have a high quality data. After that, when the imagery is captured, you use a software to stitch them, so to locate them in the correct position and one after each other. Here in this case is a guess of a metashape, and based on the different images that you have over the entire field, you can have a big picture of the field, so an orthophoto, and also build a 3D model to extract then a digital elevation model that provides us information related to height. So these flights can be done with different sensors. Um, Today we will present you two case studies. One with an RGB sensor will be my case study about the characterization of growth of soybean using an RGB imagery. And my colleague Tom de Suave will present a case study that uses a thermal sensor, and it's related to drought tolerance. So as you can see in the image, you can, depends of the region that you want to have in your images, so the spectral information, you can select one or another sensor that covers a specific range in the electromagnetic spectrum. So I will start with my case study, which uses an RGB imagery, so that records the information in three bands, red, green, and blue. Why are we interested in this sensor? It's because it provides high detailed information. So we have really a lot of pixels per image. So that allows us to even count individual plants. And as I mentioned, it gives us three bands, red, green, and blue, that provides different spectral information, as you can see. But how can we use those bands? We can combine them as a vegetation index to, for example, to highlight the vegetation part of the imagery and only focus on those pixels that correspond to vegetation. And we can also extract the canopy height of the crop of every single flight that we do over the field. So with this two type of information, we can then go to the characterization of the growth of soybean. So just as a short introduction, soybean is originated in China and now the main production is in America. And what do we want to achieve is the adaptation to our Belgian climates. And how is this done? So the breeders evaluate the crop or the different genotypes along the growing season. They have a look to the emergence, so how well the crop established at the beginning of the season, then how fast the crop covered the soil, and also, for example, they score maturity, so when the maturity stage is achieved. So we want to do something similar, but with imagery. So if we fly at the beginning of the season, we can have a look to the percentage of emergence. Also, then when the canopy is closed, we can measure the maximum height reached. Or at the end, with the last flights in the growing season, we can see how fast the crop senescence, how fast the leaves turn yellow and drop. So as we fly at different moment of the growing season with all those points, we can then fit a curve and extract some relevant information from this curve. And as we can cover the entire field trial, so every single plot, we have such a curve for every plot established in the field. So here it's an example of our breeding trial, and the different curves allows us to compare compare the genotypes and make a selection. What type of phenotypic variables can we derive from the curve? Here is related to height. So we measure the height in 14 different points, and we fit the curve. So parameters that can we extract. For example, the maximum growth rate, and also when this maximum growth rate happens. We express it in thermal time because we convert the day of year to thermal time, as temperature is one of the main driver of crop growth. We can also derive the maximum height, and when this maximum height is reached, also in thermal time, we can also derive parameters such as the determinacy, how long the crop grows, and also when we take into consideration the last flights done in the season, we can calculate the senescence rate, so how fast the leaves turn yellow and fall. So here it's in terms of height. But we can also do something similar with, for example, the canopy cover. So how fast the crop covers the soil, and we can also derive some biological parameters that we can link to traits like first coverage, how successful was the establishment. We can also calculate growth rates, etc. And how can we use those parameters? We can apply them to selection. We can compare the genotypes based on three parameters. For example, here, the maximum height, the early vigor, and the senescence. So we take two crops that have a difference in early vigor. One covers the soil faster than the other, but the one that covers faster has a lower height than the one that has a less coverage. We can also have a look then to senescence rate, because, for example, those two genotypes have a similar early vigor, so they cover the soil more or less at similar rhythm. They have a slight difference in height, but then in senescence rates, they behave completely different. So the one on the top turns the leaves yellow way faster and lose them. So we can really have an overview of every genotype established from the beginning to the end to compare them to apply to selection. So it's a really robust tool for breeders to use it for their selection. So I would like to give you some take-home messages to end this presentation. So I would like to remind you that the RGB sensor provides high-resolution data and valuable, that this methodology deliver robust and objective phenotypic information, and really they can be linked to biological traits, that we can screen a larger number of plots using a UAV, and this contributes to a more efficient data collection. And really important, this method has been developed in close collaboration with our breeders, and it's implemented in our breeding programs. So with this, I would like to thank you for your attention, and I would also like to thank my colleagues, because I don't work alone, it's a teamwork. And if you have further questions, you can always contact us. Thank you.